A remarkable feature of many plasmid is the ability to transfer themselves and other DNA elements from one cell to another. This process is called conjugation. This conjugation process first discovered in 1947 by Joshua Lederberg and Edward Totham. They found that mixing some strains of E. coli with others resulted in strains that were genetically unlike either of the originals. Lederberg and Totham suspected that bacteria of the two strains exchanged DNA. That is, two parental strains mated to produce progeny unlike themselves, but with characteristics of both parents. However, at that time, plasmids were unknown. Many naturally occurring plasmids can transfer themselves and are said to be self-transmissible. Self-transmissible plasmids encode all the functions they need to move among cells, and sometimes they also help in transfer of mobilizable plasmids. Mobilizable plasmids encode some but not all of the proteins required for transfer and consequently need the help of self-transmissible plasmids to move. Any bacterium harboring a self-transmissible plasmid is a potential donor because it can transfer DNA to other bacteria. Any gram-negative bacteria such cells produce a structure called sex pillars, which is facilitate the conjugation procedure. Bacteria that lacks self-transmissible plasmids are potential recipients. Self-transmissible plasmids exist in all types of bacteria, but those that have been studied most extensively are from the gram-negative genera, that is Escherichia and Pseudomonas, and the gram-positive genera Streptococcus, Bacillus, Streptomyces. Conjugation and gene transfer in E. coli are driven by circular DNA plasmid called the fertility factor or sex factor, which found in some cells but not all cells. This is a simplified view of conjugation by a self-transmissible plasmid, the A plasmid. A replica of the plasmid is transferred from the donor to the recipient cell so that both the donor and recipient cells have the plasmid. A cell that has received the plasmid by conjugation is now known as transconjugant. Hence, to understand how to make a cross in E. coli, we have to understand the properties of F. Cells carrying the F plasmids are designated F plus and those lacking it are F minus. The plasmid contains approximately 100 genes which give the plasmid several important properties. Like all other plasmids, the F plasmid can replicate its own DNA, allowing the plasmid to be maintained in a dividing cell population, which is seen in the figure A. Cells carrying the plasmid promote the synthesis of pili on bacterial cell surface. Pili are minute proteinaceous tube-like structure that allow the F plus cells to attach to other cells and maintain contact with them, that is to conjugate. Now, F plus and F minus cells can conjugate. When conjugation occurs, the F plus cells can act as F donors. The F plasmid DNA replicates and the newly synthesized copy of circular F plasmid molecule is transferred to the F minus recipients. However, a copy of plasmid always remains behind the donor cells. The recipient cell becomes converted into F plus because it now contains a circular F plasmid. The transfer of the F plasmid from F plus to F minus is rapid, so the F plasmid can spread like wildfire throughout a population from strain to strain. F plus cells are usually inhibited from making contact with other A plus cells. Therefore, the A plasmid is not transferred from A plus to A plus. Sometimes, if plasmid carries within its genome one or more insertion sequence elements. An IS elements, that is the insertion sequence element, is a mobilizable segment of DNA that moves from one place to another within the host chromosome or between chromosome and plasmid. The existence of a specific IS element both in the plasmid and in the chromosome facilitates the homologous recombination. This homologous recombination between the two circular DNA leads to the integration of plasmid into the bacterial chromosome. When this occurs, F can drive the transfer of the entire host chromosome into the recipient cell along with its own integrated fDNA, which is shown in the left top figure. This transfer is detectable because donor and recipient alleles recombine to produce genetic recombinants, and that can be identified. This genetic recombination is shown in the below figure. It is possible to isolate the rare cells in which the F factor integrated into the host chromosome from the bacterial population. So in comparison to this, cells can donate chromosomal alleles during F transfer. So the frequency of the recombinants for these cells is much higher than it is for the cells in an original population where F factor is not integrated in most cells. Therefore, strains with an integrated F factor are termed high frequency of recombination strains to distinguish them from normal F plus strains. Now, the integrated F factor occasionally leaves the chromosome of an HFR cell and moves back to the cytoplasm. In some rare cases, carrying a few host chromosome genes along with it, which is shown in the blue color. This modified F, called F prime, and can transfer the specific host genes to a recipient F minus cells in the same way that F is spread. Thus, the recipient cell now contains two copies of the same gene, one resident copy on its bacterial chromosome and one copy on the newly transferred cytoplasmic F prime factor. Now, the whole conjugation process is a complicated process that requires the product of many genes. The genes required for transfer are called the TRA genes, which include at least 20 genes. In addition to the TRA genes, or a T sequence is required for transfer. The TRA genes of a self-transmissible plasmid can be divided into two components, the DTR component for DNA transfer and replication and MPF component for mating pair formation. MPF components encode for large membrane-associated protein structure, includes the pilus that is responsible for holding and mating cells together and the channel between the mating cells through which the plasmid is transferred. In addition to the pilus, the MPF system encodes a channel or pore through which DNA passes during conjugation. So the MPF component is the first to make contact with the recipient cell. Then the information is communicated to the DTR component before DNA transfer occurs. The communication between the DTR and APF system is provided by proteins called coupling proteins, which are part of the MPF system. These coupling proteins provide the specificity for the transport process so that only some plasmids are transferred. The DTR component of a self-transmitted plasmid is involved in preparing the plasmid DNA for transfer. A central part of DTR component of plasmid is the relaxase. This is a specific DNA endonuclease which makes a single-stranded break or nick at the specific site called the nick site or in the ORT sequence to initiate the transfer process. It also recycles the plasmid after transfer. The relaxase breaks the phosphodiester bond at the nick site by transferring bond from a deoxyribonucleotide to one of its own tyrosine residue. Now the relaxase protein bound to the 5' end of the nick through its tyrosine, and the relaxase protein is transferred into the recipient cell along with the DNA. In fact, it is probably the relaxase protein itself that is transferred and the DNA just goes along for the ride. Once in the recipient cell, the relaxase recyclizes the plasmid by doing essentially the reverse of what it had done in the donor cell. It binds to the two halves of the cleaved ORT sequence, holding them together while it transfers the phosphate bond from its tyrosine back to the 3' hydroxyl group of deoxyribonucleotides in the DNA. This trans-esterification reaction reseals the nick in the DNA and releases the relaxase, which is then degraded. The transferred plasmid is now a single-stranded circle in the recipient cell and is subsequently replicated by the host cell's replication machinery. Plasmids are not the only DNA elements in bacteria that are capable of transferring themselves by conjugation. There are numerous examples of other elements often called integrating conjugative elements that are normally integrated into the chromosome and do not exist autonomously like plasmids. For example, TN916 from Enterococcus faecalis can transfer from the chromosome of one bacterium to the chromosome of another bacterium without transferring chromosomal genes. Now, this is the summary of the total conjugation procedure. Okay, thank you. That's it.